All right, we might get going and see people are still trickling in. So I'll give my little spiel at the beginning and we'll go from there. I assume everybody can hear me all right and see my screen. If not, shout out and I'll adjust something. So thank you for joining our April meeting of the Portland Urban Beekeepers. We are here tonight to hear Kelly Kulinek, maybe something like that. Close enough for right now.
Yeah, it's Kohlhanick. It's a tricky one, I know.
Kulinek,
Yeah, thank you.
from Washington State University. And yeah, so let me go through my little PowerPoint. Our little shameless plug, please consider joining a pub if you're not a member already. We do have some great benefits for those who join, primarily our swarm list and our Bee Buddy program. We also have our bee days at the apiary where you get a lot of hands-on beekeeping experience alongside other beginning beekeepers and led by some pretty excellent mentors. So that's always really fun. We've had a few of them already. And we also have our library with a bunch of resources as far as books and candle molds and stuff like that. And then our equipment rental, which comes in handy, especially during the honey harvest season, where we have our extractors, both manual and mechanical. And also you can post on our website, and most importantly, it helps fund our mission to help fund bee research. So yeah, it's only $25 for a year and you get a bunch of great stuff and access to a lot of friendly people. Like I mentioned, the Bee Buddy program is one of our main benefits. So that's where we try to partner up people who want to learn and people who are willing to mentor others in their neighborhood. So it's really easy. You just go to the pub website, you can click on the resources tab and then fill out some information about either what you're willing to mentor people on or what you're looking to learn about. And then you'll get a little pin on our map. And once you're there, you can see who's around you and click and contact those people and make some connections. I've definitely met people in my neighborhood that way, and I think it's pretty awesome. We mentioned this last month, but I just wanted to make sure people didn't feel like they were getting left out of the loop, but we did upgrade the Swarm software. So if you have not re-signed up since the last meeting, so 4 weeks ago, you still need to sign up to get onto the new Swarm software. So we have had calls coming in. I meant to check before this meeting, but I would say we've had a dozen or so, maybe even more of swarms reported so far. And we're definitely getting into swarm season coming up. So how do you do that? It's pretty easy. All you have to do is go back to the pub website, and I will drop a link in chat in a moment for that. So you can just link directly to the swarm register, and you'll just fill out your information. You'll fill out the registration code, which links you to us and not to another beekeeping club. So you'll get the swarms reported in your area. And the most important piece on this is actually the range. So if you have any questions, because I'm going too fast, or you don't want to ask in front of the group, feel free to email Jana. But I wanted to talk to you about the range on this kind of stuff. So the range dictates how far you want to go, and actually will dictate if you get called for a swarm or not, or put first in that swarm list. So if this is the range of person A, and even though they're position 11 on the swarm list, so we have, incrementally people in like a row or like an Excel sheet. And if the B is within their range, then, and the only other person on that list does not have a range that intersects with them, then they would get the text first to get have a chance to get the bees, right? So that can kind of make sense, right? So if the swarm is within your range and you're the highest number within there, then you're going to get the text first and the opportunity to respond. If the scenario is that you both have an overlapping range and the bee swarm is within there, then obviously the higher position would win out as far as the first text message that goes out. That doesn't mean they get to claim it over everybody else. It just means they have the opportunity to claim it first. If you're worried about the range, just set the range at 99, which is the 99 miles you're willing to travel, and you will be included in every swarm call that goes out. So any questions, feel free to drop them in chat. Happy to go over them, or if you want to unmute too. All right. Another really incredible thing that we do here is the Bee School, which is led by Susie. She is on the board as the education liaison outreach superstar. And she led it last year and maybe in the year before that. And she's doing it again this year. And we're improving upon it. Every year we have done 2 of the 4 classes, and we actually use Dewey's book, Honeybee Biology, which is a pretty fantastic book as well, even if you don't go to the class. And so we've done 2 classes so far, and the next one is be coming up on, is this Monday or Sunday, one of these days. So in the next few days, we're talking about honey harvesting, robbing behavior, queen events. So trying to keep you ahead of the game as far as the beekeeping season, so you don't feel like you're playing catch up. If you missed any of the classes, they are recorded. So even if you sign up today, you will have access to these 4 hours of classes you can rewatch, and you get the chat as well on the side, so you can see everything that was going on in there. And it's still super helpful. And it also gives you access to Susie too, to answer any questions you might have in this upcoming season or upcoming Zoom session, essentially. So really great there if you're a beginning beekeeper or want to brush up on some of your skills. We have decided that the next bee day at the apiary, so when we're going to go out to Green Acres again, will be May 1st. We did push it to this upcoming weekend, hoping for better weather, but that's not going to work out. We're still looking for some bad weather this upcoming weekend. And then the next one was going to fall on Easter, and we didn't have the necessary personnel to actually staff that. So we decided that May 1st is our new goal as far as doing full inspections and getting everybody out there. And yeah, that'll be 10 a.m. to noon. I'll send out a reminder email the day or 2 before, so everybody doesn't feel like they forget about it. And it should be a lot of fun, and we're hoping for some good weather. This is Green Acres where we're going to be doing it. So we have hives in this area right now, and we have 2 Langstroths, and we're looking to populate a top bar hive out there. Also, this site is used by OSU for research of beehives. So we get to kind of see what they're doing out there, which is kind of fun. But essentially, as far as parking, because it can be a little tricky, you're going to want to park in the parking lot next door, which is actually a little boat ramp park for this park over here, and then just walk around through the entrance, and you can't miss it. You just come all the way down and you'll see a bunch of bee nerds sitting here under a shelter or mingling about in their bee clothes. So again, I'll send out a reminder when we come close to the May 1st date. All right, so that's enough of me. I'm going to pass it over to Kelly, but a little look at our upcoming speakers. We have Ramesh next month, and then we have our very own Dewey in June, which will be really exciting, talking about queens. And then in July, we have Cassie from Oregon State Extension talking about the Asian giant hornet and some of that stuff up there. So since we're in Portland, I thought that would be kind of fun to hear about the advances of that bee killer. So that is all for me. I will stop sharing and pass it over to you, Kelly.
Thanks, Ian. What a cool apiary location your club has. That's so neat, right on the water there. Okay, yeah. Hi, everyone. So as Ian said earlier, my name is Kelly Kulhanek. I am a postdoctoral researcher at Washington State University, which means that I have my PhD, but I don't have like a real big girl job yet. So kind of in between, you do postdocs for different professors. So I work for Dr. Brandon Hopkins up here at WSU. And tonight I'm going to talk a little bit about the work I did during my PhD at the University of Maryland. So just a little bit of background on me. I completed my PhD from the University of Maryland in April of 2020. So right at the beginning of the pandemic. So my defense and, my parties and everything was all over Zoom. And here we are still over Zoom 2 years later. So it's pretty crazy, but yeah, I think the Zoom thing also has allowed me to give a lot more talks to a lot more audiences than I would have if we were all still doing all in person. So that's been kind of a nice silver lining of the whole situation. And so while I was in Maryland, I did a lot of work with sort of these smaller scale beekeepers, more hobbyist beekeepers. And that's what I'm going to talk about today is these sort of best practices for small apiaries. And then, that was great. I had a lot of great experiences there. I met a lot of really great beekeepers, but I wanted a little bit more experience with commercial beekeeping at the commercial level. So I came here to WSU for my postdoc. I started in here of August of 2020. So I've been out here for about a year and a half. And here I'm doing more commercial beekeeping management practices. So my supervisor, Brandon, does a lot of these really like large scale studies, collaborating with commercial beekeepers using like dozens or hundreds or thousands of colonies for experiments. So it's definitely been a really different experience out here. I'm currently actually in a hotel room in Twin Falls, Idaho, to sample a commercial beekeeper who just got back from almonds tomorrow. So that's why I have this weird, like generic art behind me of the Snake River. But yeah, so it's been another really good experience up here. I'm originally from San Diego, California, so I'm very happy to be back on the West Coast, just a little bit closer to home than I was in Maryland. And yeah, most of the commercial practices I'm working on here have a lot to do with this concept of indoor storage. So commercial beekeepers storing their colonies indoors in these sort of temperature controlled buildings over the winter. Which is a super interesting system that I really knew nothing about before I came here. And so, yeah, it's been really cool. I've gotten to tour these facilities and like interview beekeepers who use them and make videos about it and stuff. And if anyone's interested in this is a lot of what we do at WSU, and we have a lot of resources online, mostly through Project APISM at their website. So kind of a cool thing to research. But today, like I said, I'm going to go kind of back in time a little bit. To my PhD and my time at the University of Maryland. And while I was at the University of Maryland, I did a lot of work with the Bee Informed Partnership. And I'm just going to briefly introduce what Bee Informed is in case anyone's not familiar, because a lot of what I'm going to talk about today directly has to do with Bee Informed Partnership. So Bee Informed Partnership is this sort of national nonprofit, and it's a collaboration with a whole bunch of different universities sort of all across the country. And I was lucky enough as a graduate student to be at the headquarters of the Bee Informed Partnership at University of Maryland. So my PhD advisor, Dennis Van Engelsdorp, was the president of Bee Informed at the time. And so the diagnostic lab and the headquarters and everything was run out of our lab at University of Maryland. So I had access to all these amazing collaborating universities all over the country and these huge data sets that Bee Informed has collected over the years, which I'll talk a little bit about in a minute here. So this ended up being a really cool experience for me. So we were headquartered in Maryland. We have all these collaborating universities you see on the screen. The idea here is to kind of have a collaborator in sort of these big hubs of beekeeping in the US, right? So we have this branch in California that's sort of in close proximity to the California queen breeders that are down there. We have branches in the Midwest that are close to beekeepers while they're doing honey production. In Oregon, beekeepers do a lot of seed production and pollination. In the South, there's lots of package production. So really trying to meet the majority of beekeepers where they are in the field so we can actually, meet with them, talk with them, hear their challenges, and sort of directly address their needs. And so the goals of Bee Informed are to reduce colony mortality by analyzing which management practices work and which ones don't. And so, I think we all know as beekeepers that beekeeping can be a very sort of opinion-based activity. And lots of people have lots of strong opinions on the right or wrong way to do things in beekeeping. And, if you go on YouTube or Google, you'll hear people coming up with all sorts of new kind of exciting sounding, crazy ideas about how to make things easier or better. And so the goal here is to really kind of test some of those things using science and data to actually figure out which ones work well and which ones don't work as well so that we can save you all time as beekeepers and not have you have to try them out on your hives yourselves. And then also through doing that, kind of to provide a bridge between research and the beekeeper, right? So this is a very two-way street where we do a lot of talking with beekeepers, listening to the challenges, and then doing research that sort of directly meets those needs, and then handing over the results of that really quickly. So trying to really streamline this research process to make sure that the projects we're doing are actually useful. And so Be Informed has all these different programs, sort of for all different shapes and sizes of beekeepers. So the tech transfer teams are kind of the commercial beekeeping branch. These are like really highly trained individuals who actually go into commercial apiaries and sample and inspect their colonies, and then kind of give recommendations to the beekeepers based on what they see. The Sentinel Apiary program is a really cool program for mainly for more hobbyist beekeepers. It's a little bit smaller scale, and it is a colony health monitoring program. That's also really cool for clubs to do if you guys are interested. A lot of people like to do this in their club apiary. Sign up for a Sentinel Apiary where you sort of sample and inspect your colonies for 6 months, and you actually send your samples to the lab in Maryland, and then you get a report back with your Varroa and Nosema results. There is a cost to participate, but some clubs like to use some of their dues to kind of cover the cost of that. And it's a really good way to teach your new beekeepers sort of how to monitor and manage for mites all year, which is really cool. The main one of these things I'm going to talk about tonight is this National Loss and Management Survey. And so this is sort of one of the biggest, kind of most important things Be Informed does, and also kind of the thing they've been doing for the longest. And so the National Loss and Management Survey is this annual survey of US beekeepers where we ask, how many colonies did you have before winter and after winter, and before summer and after summer, so that we can calculate your colony loss rates. And we've been doing the survey since about 2007, when colony collapse disorder sort of first came on the scene. And basically, what we realized, it wasn't me back then in 2007, I was too young to be helping with any of this, but what Be Informed realized back then, when colony collapse disorder came on, we had these beekeepers really kind of panicked and saying that, way more of their colonies were dying than normal, and something really crazy was going on. And we realized that we didn't have a very good way of documenting how many colonies were dying each year. So the USDA and some other sources will count the number of colonies in the US each year, but that's not really a good representation of the turnover rate between years, right? So we know as beekeepers that you can lose a lot of colonies over winter and have the same number in your apiary from year to year, because you make splits, you buy new colonies, you kind of recoup those losses. And just counting the raw number of colonies in the United States doesn't really capture that turnover rate, which can be very stressful, right? Economically and emotionally for beekeepers to endure such high losses. So that's kind of the idea behind this loss survey, is to really picture what that turnover rate looks like. This is last year's results. We had about a 30% winter loss, which is right around average. I did just want to mention that the survey is open now. It comes out on April 1st each year and will be open until April 30th. And anyone can go on and take the survey. So if you're interested in contributing to this research, you can go to this website here. It is super important research. It's really the only survey of its kind, and people cite the results of this survey constantly in scientific papers. And when we write grant proposals, to really stress to the funding agencies how urgent it is that we get funding to study bees, we use these survey results to sort of help justify the need. So anyone who takes the survey, we really appreciate it. And it's open now. So if you have some spare time, go ahead and take it. So that's kind of the loss survey component, right? Where we're just asking about the number of colonies you have and calculating your loss rate. And then we also have the other component of the survey, which is the management survey. And so this is the component of the survey that's really lengthy. It's a pretty long survey. And that's because we're trying to capture a really high level of detail, right? So, this says that we're asking about 72 different criteria from 8 different domains. It really can be more questions than 72, kind of depending on how you answer. But basically, we just want to know every single management practice you used in your apiaries in the last year. So we're asking, if you treated for Varroa, what did you use, what dose, how often, what months, if you monitored, if you treated for other things besides Varroa, what you fed, how long you've been a beekeeper, all things like this. And so the management survey is really interesting because we can kind of track how beekeeping management practices change over time in the United States, right? So this is a really good example. This is just whether or not a beekeeper used Varroa treatment. And so, we're just looking at the number of responses, basically the percentage of people who responded that they did use Varroa treatment in blue or did not use Varroa treatment in gray. And this is referring to any Varroa treatment, chemical or non-chemical. And this is over the whole life of the survey, right? So all the years of the survey back to 2007 combined, you can see that less than 50% of beekeepers who have taken the survey over its whole life have used Varroa treatment in the year they took the survey. And so, I think we know now in 2022 that the vast majority of people are managing for Varroa in some way. But this is kind of a good example of how these things change over time and why it's interesting to keep track of them because, as different issues emerge, it's good to know sort of how we're responding to them as beekeepers with our management. And then the other really interesting thing we can do, of course, is we can combine the loss survey and the management survey, and we can correlate mortality to management. So we can look at your management practices and look at how many colonies you lost. And we can say like, oh, wow, seems like beekeepers who use practice A do really well and lose very few colonies compared to beekeepers who use this other practice seem to not do as well. And so again, Varroa treatment's a really straightforward example. Here we're looking at the average winter loss per beekeeper. So, we're just looking at the percentage of colonies they lost over the winter. So, lower is better here, of course. And that top bar is beekeepers who used Varroa treatment, who lost on average around 34% of their colonies compared to beekeepers who don't use Varroa treatment, losing about 43% of their colonies. And again, this is all years of the survey combined. But, based on this, we can say there's this pretty consistent effect where beekeepers who use Varroa treatment lose fewer colonies than beekeepers who don't. And so we can now use this as sort of the basis for like a theoretical best management practice that we could maybe start to recommend that if you want to improve your chances at overwintering success, you might want to consider using Varroa treatment. And you'll notice I'm saying like may and might a lot because as scientists, we never want to like make guarantees or say anything's 100%. So you'll hear me using like a lot of qualifiers. But this is a pretty consistent result we feel pretty good about. And so, we can do that for Varroa treatment, and we can also do that for every single question we ask about on the survey, right? And so we could go through all 72 of those criteria on the survey and look at which management practices are associated with the lowest colony loss. And then we could hand you a list of 72 management recommendations we think you should try. But that's not the most helpful, right? 72 things to keep track of is a lot, kind of more than any one person wants to take on. And so we want to look at how can we prioritize which of those management practices are most important. And so it turns out that not every management practice we do has a huge impact on winter loss. In some areas of beekeeping, it doesn't really matter what you do. It's not going to make that big of a difference for winter loss. So we kind of want to look at which of these management practices are kind of going to give you the most bang for your buck. Or, in other words, if I was going to come to your apiary and look at your bees and give you like your top 3 most important things I think you should focus on that are going to give you the biggest improvement in your winter loss, what are those things going to be? And so my colleague at University of Maryland, Natalie Steinhauer, did a lot of this work for her PhD. She was finishing her PhD right as I came into the lab in 2016. And so she kind of went through, all the tens of thousands of survey responses from the loss and management survey and did a lot of really fancy statistical analysis to sort of narrow down which of these practices are really important and have a really big impact on winter loss. And she did this for different groups of beekeepers. So she kind of broke it down into backyard beekeepers and professional beekeepers, and then even further into backyard beekeepers in the north and south, and professional beekeepers that are stationary and migratory. And she did this because, if we're thinking about making recommendations for management practices, the baseline practices that a migratory professional beekeeper needs to use are very different from the practices that a lot of backyard beekeepers need to use. Their goals for as an operation are different, their challenges are different. And so we want to make sure that these recommendations are as specifically tailored as possible. And so basically how Natalie did this is make this really scary graph where she ranked all 72 of those management practices, from 1 to 72. And this is based on their impact on overwinter mortality. So they're ranked in order based on how important they are at predicting your overwinter loss. And so basically, stuff that's ranked really highly, from 1 to 10 is very important for winter loss, has a really strong, consistent impact on overwinter mortality. And then these practices that are sort of at the back here, ranked 60 or higher, it turns out that no matter what you do in that management practice category, it didn't have that much of an impact on the overwinter mortality. So no particular practice in any of those categories seemed to be that much better than any of the other practices. And so if we're going to start thinking about like a basis for, recommendations we think you should focus on, we're going to start with this stuff down here that's ranked super highly as having a really big impact on winter loss. And so what each of those things are for those categories I mentioned are now on the screen. And, one of the first things that kind of jumps out at you is that there's kind of a good amount of overlap. But I just want to start with the professional beekeepers here. And so basically, the interesting thing about the professional beekeepers is that there was a lot of variation in their answers. So basically, the correlations between management and mortality were not as consistent, were not as tight or strong. And so, it's a little bit harder to make these like broad, generalized recommendations for these professional beekeepers. And so because there's so much variation between operations and because they need things to be so specifically tailored to like, where their bees are at any given day of the month, they really need more one-on-one recommendations. And so these kind of more general management recommendations that we can use survey data for seem to not apply as well in a professional beekeeping setting. But for backyard beekeepers, these correlations were super, super strong. And there were really strong associations between some of these management practices and winter loss. And so, you'll also notice that these are the top 5 most important things in both these categories of practices. And 4 out of the 5 are the same in northern backyard and southern backyard apiaries. So for all backyard beekeepers in the United States, and for our purposes, a backyard beekeeper is anyone with 50 or fewer colonies. I don't know a lot of people who can fit 50 colonies in their backyard, but that's the cutoff we use. And so you can see that for pretty much any backyard beekeeper in the United States, these top 4 management practices are the same and were the most important for their impact on winter loss. And so, now we can sort of use these as a basis for thinking about management recommendations. And what we can do is compare the average beekeeper to the theoretical best beekeeper in each one of these categories. And I'm going to walk through each one of these categories in a minute, but I just want to clarify by what I mean by average. So here, the average beekeeper is sort of the most common response to this question on the survey. So the most frequent practice used by hobbyist beekeepers in each of these categories. And then that theoretical best practice is the practice that's associated with the lowest winter loss. And so I'll walk through each of these, starting with dead outs. And so, here what we're talking about is the action you take when you find a dead colony. So you arrive at your apiary, you find a dead colony. What do you do with that equipment, those boxes and those frames from that dead out? And so, here again, we're looking at the average winter loss per beekeeper. We don't have that many options of things we're going to do with that equipment, right? The most typical action to take in this situation is that you're going to store that equipment for later use. right? You're going to take those boxes and those frames and put them in your garage or your shed. And you're just going to store them in there probably until the following spring when you buy a new package or nuke to replace that dead colony, right? So this is, definitely the most common response, but you can see that it's also the response that's associated with the highest level of winter loss. So beekeepers who store their dead out equipment lose about 49% of their colonies on average, compared to all the way down here, beekeepers who immediately replace that dead colony lose about 10 percentage points fewer, only 39% of their colonies. So that's the theoretical best practice in this situation. And so to immediately replace that dead out means to, replace it with a new colony. Usually that's accomplished by performing a split and using that dead out equipment for a new split. And, this one's pretty weird, and we can't really explain like why this came up as so important. And the reason it's kind of weird is because you think about the timing and the time of year where you're going to be finding dead colonies in your apiary. It's not going to be the same time of year where you're needing to make splits and to immediately replace colonies, right? So this one is pretty tricky to actually do in practice. And so at this point, I just kind of like to give the little disclaimer of like, just, if you have the opportunity to try this in your apiary, if for some reason you find a dead out and you have the opportunity to immediately reuse that equipment for some reason, we just recommend you give it a try. And that's, really all I can say about this one. And that kind of goes for all of these things, is that, if you're already doing something in your apiaries that works really well, then you should just keep doing that. But if you have an opportunity and you want to try something new, the data suggests that these things are associated with lower loss and came up as super, super important. So again, we're not really sure why this works. I would like to eventually get back into doing more research about this and kind of try to understand what the mechanism is behind this and why it came up as so important. But for the purposes of this study, we either stored that equipment for later or tried to immediately replace those colonies. And then the second thing that came up as super important is how you get your new colonies or your little baby bees each spring, right? So we have several options of how to obtain new colonies each spring. The typical backyard beekeeper is buying packages, which have a relatively high mortality rate, right? And so the theoretical best practice here is to either split your own colonies. You can see it has a much lower level of loss here, is to split your own colonies. Or you can see that purchasing nukes has a very similar loss rate. So if you don't have enough colonies available to perform splits, then we recommend you buy a nuke over a package. And so, in my experience, I think part of the reason that this mortality rate on packages is so high is because a lot of beekeepers in their first year get really excited and they buy a couple packages. Say you buy 2 packages, right? Your first year of beekeeping, and say you're a brand new beekeeper, so you do not have any wax combs drawn out, right? You install those packages on 10 or 20 frames of bare plastic foundation. And then those bees have to put in a lot of labor to draw out all that wax in that first year, right? It takes a lot of energy for bees to draw out that much wax. And so, I think part of what happens in people's first year beekeeping is that that first year really kind of ends up just being an investment in that wax, right? And so the colonies might spend a lot of energy drawing out all that wax and then don't have a lot of energy left over to store honey or to prepare for overwintering. And so they might not end up surviving the winter, but you have a good investment in the form of all those beautiful wax combs. And now when you go to install more packages next spring, you're starting way ahead of where you started last year. So, and again, if you think about like an average new beekeeper buying 2 packages, if you lose 1 out of 2, that's 50% loss, right? So I think that's part of why this loss rate on packages is so high. But if you buy those nukes, they come with wax frames already drawn out, and they're just like a little bit ahead of the packages usually, with the queen already in there laying and everything. So we think the success rate is just a little bit higher with nukes, but you can still be fine with packages. I know this time of year, I feel bad like telling people not to buy packages because usually people have already bought them. So enjoy installing your packages. It's like my favorite my favorite day of beekeeping every year is package install day. Okay, and then the third practice that came up is super important is this idea of how you treat old brood comb, right? So if you've been a beekeeper for a few years, you're going to have wax combs that have sort of built up and that you have probably been storing for a little bit of time. So say, you have a dead colony that died, in February, and then you go to store that equipment with those brood combs and wax frames and everything in there. And, this is kind of an example of really old comb versus really bright, new, beautiful comb. But the idea here is, do you treat that old brood comb in any way before you reintroduce it into a new colony? And so there are a couple of different options of how you can treat your brood comb. And most of these, really the idea behind this is just a hygienic practice where you want to make sure that you clean out the wax in between it being in a dead colony and it going into a new colony, just to make sure that it's like not passing anything weird into the new colony. And so there's people irradiate their combs, fumigate them, do other chemical treatments. The average beekeeper is not treating that brood comb in any way before reusing it. And you can see that not reusing the comb at all. So basically never reusing old brood comb, just like throwing it all away, has the lowest level of loss, but it's super, super similar to freezing. And so, we don't want to not be able to reuse any of our brood combs because, like I said, it's a big investment for the bees to draw those out. So we want to be able to reuse them. And so it seems like, if you freeze those combs before you put them in storage or introduce them to a new colony, that is a pretty good hygienic practice that works just as well as not reusing the comb at all, which is really cool. And so, but, by freezing here, we're generally just talking about like your normal freezer temperature of like your kitchen freezer or a chest freezer that you might have in your basement. And we try to freeze for a minimum of 24 hours. And so, we also have collaborators in Minnesota who leave their frames outside all winter and they're frozen solid for 6 weeks. And so, that counts also. But just this idea of kind of putting them in the freezer to kill anything residual that might be living in there. This is great too, just for things like wax moth. And I know we don't have small hive beetle over here, but we did in Maryland. It was very important for small hive beetle for us. And I think there's also some benefits for bacteria and viruses also, just to kind of clean that comb out. And then, of course, the fourth thing that came up is super important is Varroa treatment. And I don't mean like treating your sick Varroa mites. I mean treating your bees for Varroa. And again, of course, beekeepers who use Varroa treatment lose fewer colonies than beekeepers who don't. But the actual average practice here is to treat your colonies 1 time per year in the fall. So the average hobbyist beekeeper treats once per year in August or September. And I actually think the frequency of this has gone up. When we originally did this analysis, it was like 2015, 2016. And at that time, it was only 1 time per year in the fall. But now I think most people actually treat more often than that. But the theoretical best practice here is to monitor these colonies monthly and to treat whenever you exceed this treatment threshold of 3 mites per 100 bees. And this is sort of this generalized treatment threshold we use of 3 mites per 100 bees. Again, I think that could probably be tweaked depending on your area or the time of year. Like 3 mites per 100 bees now, I think would be pretty high to be starting the year that high. So you might maybe want to treat even if you had it a little bit lower than that before you put supers on. But use your local club and your local resources for the best information about that. But so now basically what we've done is sort of walked through, these 4 theoretical best practices and these 4 average practices. And so we sort of have these 2 different management practice regimens where we have a whole average practice regimen and a whole best practice regimen. And now what we can do is actually test this on colonies in the field to see if these things actually make a difference, right? So, again, I'm always saying like may or might help. And so really the best way to see if these things actually improve colony health and survival is to test them on colonies. And so that's what I did for the first 3 years of my PhD, was do a 3-year field study, sort of testing these average practices against these theoretical best practices. And I will just point out that average practices here are always in the color orange and best practices are always in the color blue, which will come back when we talk about the results of this study. But like I said, for the first big chunk of my PhD, I was doing this field study. And so, you know, I was here in Maryland. We had 3 locations in Maryland, sort of in northern, central, and southern Maryland. And then again, I was super fortunate to have collaborators with the Be Informed Partnership that did this study in different areas as well. So we had, you know, Ramesh's lab here in Oregon do a replicate of this for us out here. Marla Spivak's lab in Minnesota did a replicate. My dog's with me in this hotel room. He might bark. And then we also had collaborators in Tennessee and North Carolina do this as well. And you can see what we kind of tried to do here is split this up into these different climate zones to test whether these practices work just as well in different climates, which can have a pretty big effect on our bees and our beekeeping. And in each of these locations, we had 20 colonies. So all 7 locations had 20 colonies. And in each place, 10 of the colonies were treated according to those theoretical best practices, and 10 were treated according to those average practices. And so, you know, again, we did this for 3 years. We started in the spring of 2016 and went through the spring of 2019. And for each of those 3 years, we were sort of actively sampling and inspecting these colonies for 6 months from May to October. So they received like a monthly health inspection where we recorded queen status, brood pattern, and frames of bees, and where we also took a Varroa and Nosema sample monthly from May to October. And then seasonally, so in the spring, summer, and fall, we were also sampling for viruses. And then, of course, we were also assessing colony mortality over the summer period, the winter period, and then annually. And so I'll just jump right into results here. Basically, you know, we did not see any differences between best and average apiaries in queen status, brood pattern, frames of bees, or Nosema. So in both groups, the queens did similarly well. They laid similarly well. The brood, you know, was of similar health. And the adult bee population sizes were not very different either, which is sort of interesting. You know, you would think that managing with these best practices might lead to like bigger colonies, but that did not seem to be the case. But even in spite of that, we did see big differences when we looked at Varroa viruses and mortality. And so I'll start with Varroa. And so what we're looking at here is just the monthly Varroa load, again, in best and average apiaries. And so best is in blue and average is in orange. And so this is just, you know, the mite load per 100 bees each month from May to October. We have that treatment threshold of 3 mites per 100 bees in red, just kind of for reference. And you can see that each month of the year, the average apiaries have a little bit higher mite load than the best apiaries. And, you know, that's to be expected. We were treating the best apiaries a lot more frequently than the average apiaries. So it makes sense that their mite load's a little bit lower. One thing you will notice is that in October, those mite loads kind of even out, right? So the mite load in the best apiaries kind of catches up to that mite load in the average apiaries. And, you know, I think this kind of has to do with how closely the apiaries were spaced together. And I'll talk more about this for the second half of the talk. But basically, you know, I think there was some drifting between these colonies. And so I think the best apiaries got a few mites from the average apiaries. But the important thing to note here is that if those best apiaries were exceeding that threshold of 3 mites per 100 bees in October, they would receive a critical pre-winter treatment. So because they're above threshold in October, they got an oxalic acid drip treatment before they overwintered to bring that mite load back down to a good level before those bees had to overwinter. And then the other thing to notice is that The average apiaries are starting each spring with a higher mite load on average, and they're also exceeding that treatment threshold 1 month prior to when best apiaries are exceeding it. And so basically, they're spending pretty much the whole spring and summer with a much larger population of mites. And I think, you can kind of see the implications of that when we look at the virus results. And we'll just use deformed wing virus as an example here. We all know what deformed wing virus is. And here we're just looking at prevalence. So this is just the proportion of colonies in best and average apiaries that tested positive for deformed wing virus. And you can see that in the spring, mid-season, and fall, the prevalence is pretty similar between groups, right? So pretty similar numbers of colonies are testing positive for deformed wing virus in best and average apiaries. But when we look at how severe that infection gets, or sort of the intensity of that infection, which we use the copy numbers for, which is basically the abundance of that virus in the sample, you can see that by fall, in average apiaries, the abundance of that virus is much, much higher. And so basically, even though deformed wing virus is present in the same number of colonies in both apiaries, the severity of that infection in average apiaries is sort of much more intense. And I think that's because there are so many more mites in those colonies for so much longer of the season. That's a lot more sort of potential for vectoring these viruses around for a longer period of time. And, this is, of course, like a really big issue, right? Because once you have a high viral load, you can't treat for viruses. And so it's kind of too late to correct this at this point. And these bees are going to have a hard time making it through the winter. So really important to be proactive. And now's the time, to really get on top of your mite management early in the season and make sure you keep those numbers low throughout the whole season and just make it easier on your bees. And we saw the same trend with similar prevalence, but much more severe infections for acute bee paralysis virus and for deformed wing virus B, which is also known as Varroa destructor virus. And then when we look at winter loss, there was sort of this really interesting trend. And so here we're just looking at the total percentage of colonies lost each winter from 2016 to 2018. And you can see in 2016, we had pretty similar levels of winter loss in best and average apiaries. And so, that was the first year of the experiment. And to start everybody on an even playing field, we installed all the colonies in best and average apiaries from packages on bare plastic foundation. And so I think that was like a really big determiner of their overwintering success that first year, is that everybody kind of had to overcome being installed on their bare plastic foundation. But then, there's sort of this really interesting trend over the next 2 years where the average colonies do a little bit worse each year and the best colonies do a little bit better, until in 2018, we have a 30 percentage point reduction in mortality in best apiaries. And so, this kind of suggests that there is really this cumulative effect of management over time. And that, if you think about your colonies starting the spring with higher mite loads that they carried over from last fall, or higher viral loads, those things can really add up over time. And the same goes for good management practices. You can really just get better and better over time. And I think, some of these things occur over longer timescales than we really expect. We expect to just treat for mites 1 month and have a low mite load the next, and then we're kind of good to go. But I think some of these things really do sort of accumulate over longer periods of time. So it's kind of good to think about that context when you're changing your management practices or trying something new, that it might take a little bit longer than you think to sort of see a difference. And so, this isn't groundbreaking rocket science or anything. By increasing the frequency of Varroa treatment, we reduced the Varroa population, and that reduced the fall viral infection, which in turn decreased winter mortality. And so, this was a pretty interesting result, but one of the most interesting things I think that came out of this was this idea of why those mite loads are increasing so much in the fall. And so if you remember, in the best apiaries, they were trending with lower mite loads all summer. And then in October, they kind of spiked up real quick in the fall. And so, I think basically what happened is those colonies, the best apiaries and the average apiaries were about 50 yards apart from each other. We wanted them to be close enough together to where they would be experimentally similar. We didn't want anybody having like access to better forage or having more sunlight or less wind or anything. So they had to be pretty close together. But that also has, other implications for things like drift and robbing and this idea of horizontal transmission of mites. And so, what we're talking about with horizontal transmission is the transmission of mites between colonies and apiaries sort of across the landscape. And so, we saw that result in the best management practices experiment. And we also have seen some similar kind of weird results from things like the Sentinel Apiary program. And so for another chapter of my PhD, I kind of wanted to look at this idea of horizontal transmission of mites and, colonies spreading mites to each other across the landscape, and kind of try to understand to what scale this might be happening and if it's contributing to those really big increases in mites we see in the fall. And so, with horizontal transmission, there are kind of these two general hypotheses about how this happens. And the first one is sort of this robbing idea, where robbing bees pick up mites from high mite colonies. And so the idea here is that, we have a receiver colony and say, this is your colony, the receiver colony, and you've been a really great beekeeper all year. You've been really on top of your mite management and your feeding and everything. And so come September, you have really big, robust, healthy, strong colonies. But say one of your neighbors, for whatever reason, was not as on top of it, they might have a colony that has a really high mite load. And so your big, beautiful, healthy bees from your receiver colony are going to go out into the landscape. Usually there's not a lot of forage around in September, October. And so, bees will rob each other. And so likely they will target colonies that are a little bit weaker, maybe because they have a high mite load or for another reason. And while they're robbing this high mite colony, they will pick up a mite and return home with that mite, bringing it into the receiver colony. So even though you've done a great job as a beekeeper, your bees could be picking up mites from your neighbors and bringing them home, making it much harder for your mite treatments to work and making it so you have to repeatedly treat, and it can be really frustrating. So this is sort of one idea about the direction of how this goes. And then the other idea is sort of this idea of the mite bomb. And the idea of this one is that, this high mite colony starts to collapse under the pressure of such a high mite load. And the bees from this crashing high mite colony will not just stay there and die in the colony, but will actually emigrate from their colony and go into the landscape and sort of drift or through whatever mechanism end up in your receiver colony. And they'll sort of carry the mites from their colony with them during that process. And kind of the basis for this idea is that it's been shown in quite a few different host-parasite relationships that the parasites have mechanisms to alter the host behavior to increase dispersal. So basically, the idea here is that the mites can sense that they've sucked all the life out of this colony and they know that they need a new host. And so they have mechanisms, and this could be through viruses or through manipulating their hormones or even their cuticle chemicals that make them more likely to leave their colony and find another colony and to aid in dispersing of these mites to a new host. And so there have been some really good studies on this phenomenon, but it involves tracking individual bees across the landscape, right? So most of the studies have been done on a pretty small number of colonies over kind of a small amount of distance. And so I wanted to test this in sort of what I considered to be a more field realistic situation where we had several colonies and several apiaries kind of across a little bit bigger of a distance to see what this looked like at sort of a little bit bigger scale. And so, to test this idea of this horizontal transmission, I set up 9 experimental apiaries at one of our research farms in Maryland. And so you can see the central apiary here in yellow is called the donor apiary. And then it's surrounded by 8 receiver apiaries. And each of these apiaries has 4 colonies in it. And in the donor apiary, there are 2 high mite colonies and 2 low mite colonies. And so the idea here is to sort of put high and low mite colonies right in the center of the landscape, and then track where the bees from the donor apiaries end up. So we can search for them in receiver apiaries to kind of see, if it's more likely for high mite bees that have a lot of mites to end up in other colonies, or for low mite bees that are really healthy and engaging in a lot of robbing to end up in receiver apiaries. And so to do this, we had to paint a whole lot of bees. So we tried to paint as many of the bees in the donor apiary as possible. And so basically how we did this, these colonies were in a deep and a medium. And so I pretty much shook every frame of bees into like a plastic tote with a lid. And then we had this little gadget that is actually like a plastic cylinder that you can scoop bees into and then inject carbon dioxide in there to anesthetize them so they fall asleep. And we do this in about batches of 500. And so basically, you scoop up about 500 bees, knock them out with CO2, spread them. This is just actually an upside down IPM board from like a screen bottom board. Spread them out on there. They're actually hard to paint while they're knocked out because they're like on their backs. But as they come to, they'll actually flip over back onto their feet. But they're kind of confused and immobile for a few seconds. So it's just long enough to paint a whole bunch of them super quickly. This is actually my husband in this video, rapid painting a whole bunch of these bees for me. I drug them out here on a Saturday to help me with this experiment. Yeah, we did this several times for each of the donor colonies. The high mite donor colonies were painted red and the low mite donor colonies were painted blue. And we were also able to tell individual bees apart, which was really cool. And this is really mainly due to the fact that our painting skills were not super accurate. So this, is an example of a low mite donor bee. You can see she's painted blue. And this is actually at a receiver apiary. This photo is taken, and I'll show you how we did that in just a second. But her mark looks pretty good, right? I mean, that's more or less in the middle of her thorax. It's more or less circular. And then, we can tell that this is clearly a different individual because she has kind of a cute little heart-shaped paint mark. And then this poor bee got paint on her head and her thorax. So we can tell that she's also a unique individual. And, again, just when we were like rapid fire painting that many, our accuracy wasn't exactly as high as you'd want it for if you were painting your queens or anything like that. So, but this bee did still manage to fly. This is in a different apiary than the donor apiary. So she did manage to get there even with paint right on her forehead. And then of course we have red painted individuals as well from the high mite donor colonies. And so to catch them visiting the receiver apiaries, basically what we did is put cameras on every colony in the receiver apiaries. And so the cameras were basically these little computer motherboards. They're called Raspberry Pis. If anyone's familiar, it's like a really cheap little computer motherboard that's like the size of a credit card. And you can buy little camera modules that you can attach to them. And then basically I watched a lot of YouTube videos and learned enough Python coding to tell the camera modules how to recognize our blue and red paint colors. And so basically anytime a painted bee from the donor apiary passed underneath the camera with her blue or red paint mark, the camera would detect that specific color, blue or red, and would take a photo. So we have photographical evidence of every time a donor bee visited a receiver colony, which is excellent because it meant I did not have to manually search all of these receiver apiaries and colonies for these painted bees, which was my advisor's original idea until I told him there were not physically enough hours in the day for me to do that. So I had to come up with another solution. And then The other component we added to the receiver apiaries are these screens. And so, here you can see what the little cameras look like. Each of these little colonies has a camera right over the entrance. And so when the bees pass under the camera to go into the colony entrance, they get their photo taken. And then you can also see we put robbing screens on 2 out of the 4 colonies in each receiver apiary. And so, the idea of the robbing screen is that it will, it should prevent bees from other colonies from getting in, right? And so, we're tracking where the donor bees are ending up, but we're also tracking the changes in mite loads in these receiver apiaries and colonies. And so, basically the idea with the robbing screen is that if it keeps out the donor bees or, other visiting, robbing, drifting bees, even from other receiver apiaries, we hypothesize that the robbing screens might help slow the increase in mite load that we see in the fall. If all of this is associated with lots of visitation and mixing of bees between colonies, then robbing screens might be kind of a good intervention we could use to kind of help protect our colonies from the spreading of mites around in the fall. So we wanted to test that. So we put screens on half these colonies. Okay, and then so I'll jump right into results here. And I'm going to start with just the number of bees, the number of donor bees we found in each receiver apiary. And so we found, I think, a total, I can't do this mental math really quick, a total of, I think, like 49 donor bees. And I'll tell you, we originally painted about 16,000. So it's a really, really low recovery rate, but I didn't know if we would find any at all. So I was pretty happy with the amount we did find. And basically what you'll notice right off the bat is that the vast majority of the bees we found in receiver apiaries were from low mite colonies, not from high mite colonies. And so about 80% of the visitations were from low mite donor bees, and only about 20% were from high mite donor bees. So, already off the bat, this is kind of not really supporting the idea of this mite bomb hypothesis, where these high mite colonies are sending really large amounts of bees into the landscape to spread mites all over the place. In our study, the high mite donor colonies really didn't send out that many drifting or robbing bees at all. And then also when we look at the change in mite load in the receiver colonies, the increase in mite load was not higher in colonies that were visited by high mite donor bees. And so here we're looking at the percent change in mite load over the time of the experiment. And so, you can see that the mite loads did increase quite a bit in all colonies. And then in gray here, we have colonies that were not visited by high mite donor bees, and in red are colonies that were visited by high mite donor bees. And you can see there's no difference in the change in mite load. They both had similar increases in mite loads. So again, it seems like even the colonies that did get visits from high mite donor bees, even though there weren't very many, it didn't really increase their mite load that much. But when we look at the change in mite load of colonies that were visited by any donor bee, so here we're combining low mite donor bees and high mite donor bees, you can see that colonies that were visited by any donor bee have a much higher increase in mite load than colonies that were not visited by any donor bee. And so, in gray here, these colonies did not receive visitations from any donor colony, meaning they were like pretty isolated and didn't have any visitors, kind of, kept closed doors. And it seems like that helped them not increase their mite load as much. And then when we look at the effect the robbing screens had, that increase in mite load was also lower in screen colonies. And so, again, we're looking at percent change in mite load. The colonies that were screened are on the right here in the lighter gray color. And you can see that their increase in mite load was significantly lower compared to colonies that did not have robbing screens. And so again, seems like there's some benefit to sort of having a closed door policy and not accepting any visitors in this fall period where, spreading of mites and increases in mite loads can be pretty intense. And so just to kind of sum this up, it doesn't seem like varroa loads increase much due to high mite bee visitation or sort of this robbing idea, or sorry, this mite bomb idea. And this agrees with the other work that's been done on this phenomenon. It seems like the idea that these high mite colonies are actually sending out bees into the landscape is pretty much unsupported. And it seems like robbing is a lot stronger mechanism for this phenomenon. And the robbing screens did slow the increase in mite load. And so, this is kind of interesting when we think about that robbing hypothesis. So if I'm a receiver colony and I'm going out into the landscape and I'm robbing out other colonies with high mite loads, and I return home with a mite that I picked up during my robbing activities, I am not going to be deterred by that robbing screen, right? I'm going to pass right through that robbing screen. My nest mates will recognize me. My sisters will let me into the colony carrying that mite. And so it's interesting that the robbing screens did have an impact, because that kind of suggests that it's maybe not colonies returning home with a whole bunch of mites after they've participated in robbing. Because again, I think if that were the case, the robbing screens wouldn't have had as much of an effect. They should not have really had any effect on the mite load if these bees are just coming back to their original colony, but with mites. So that robbing hypothesis is not really super supported well by the data we found either. And so, basically, the two hypotheses we started with both kind of put the pressure or the blame for this on the colonies that are sending out bees into the landscape, and sort of blaming them for any increases in mite load. But kind of what our study showed is that the state of the receiver colony is actually probably really important as well. And so, this idea of varroa loads increasing faster by any bee visitation, so where we have low mite donor bees, high mite donor bees, bees from other receiver colonies, kind of all visiting each other, all picking up mites and visiting receiver colonies. I think there's this idea of sort of receiver colony permissiveness, right? Where how willing or able a receiver colony is to accept these visitors, these non-natal bees, seems to have a really big impact on their increases in mite loads in the fall. And so when we think about colonies that are weak or small, have low population sizes, aren't capable of defending themselves and keeping out robbers or drifters or whatever, those colonies seem to be the ones that have even higher increases in mite loads compared to stronger colonies that are able to defend themselves a bit more easily. And so, we think this idea of the robbing screen might also help these colonies be a little bit less permissive to visitors and sort of help protect them against sort of these unwanted visitations. But there's definitely a lot more work to do on this robbing screen idea. For instance, the way we set it up, we had 2 screened and 2 unscreened colonies right next to each other. So I think it's possible that, if I'm a robbing bee and I come across a screened colony, but then I look right over there and there's an unscreened one right next door, you're going to give up pretty quickly on that screened colony and just go right into that unscreened colony. It's possible they'll be a little more persistent if every colony around has a screen. So, things like that just need more testing before we really get to the bottom of how much this helps or doesn't help. But, regardless of the mechanism here, these highly infested colonies in the landscape represent a threat to other colonies nearby. And so, I think it really just emphasizes the importance of education for new beekeepers, for clubs like yours, where you're all kind of working together to make sure that your neighbors are good beekeepers, because beekeeping is a communal activity, whether you realize it or not. I think especially in urban areas where there's lots of small beekeepers packed in pretty tightly together. Everybody has to be on the same page. And so I'll just sum up really quick with sort of some overall takeaways. Basically, on that first part where we looked at the best management practices, we saw that management can reduce colony losses, but not completely, right? So there's always going to be stressors that are affecting these colonies. And even after 3 years of best management practice use, we still had, over 20% losses. And so those environmental stressors are going to kind of always have an effect no matter what. And also this idea of colony stress sort of accumulating over time, and thinking about proactive management being really, really critical. And again, like this time of year is the perfect time to be really proactive. And those classes that your club has that are also like a little bit ahead of the schedule sound really helpful as well. And mites moving between colonies in the landscape may explain some of the increases we see in the fall. So, again, just these really densely packed landscapes with lots of bees and mites. And robbing screens might help to interrupt some of this, but we have more work to do. But really, I think the biggest takeaway is that this idea of sort of cooperative landscape level varroa management is likely to become more and more important. As more and more beekeepers come onto the scene, and we just kind of all got to be on the same page together in our neighborhoods. And so with that, I will thank all of the sponsors that help support this work, all of our wonderful collaborating universities at Bee Informed Partnership. This is my WSU email address here if anybody has any more questions they want to ask later, but I'm also happy to take some questions right now.
Thank you so much, Kelly. That was very interesting.
Thank you.
Great information. Really appreciate it.
Thanks.
There are a couple questions that came through on the chat as you were going through, and I'll just kind of read them one at a time and let you address those.
Okay.
Somebody asked, can tracking strips prevent robbers from bringing mites back to the hive?
Can someone clarify what a tracking strip is for me?
Jeremiah, if you want to just unmute and clarify your question, that'd be great.
Yeah, it's a thing I tried my first year of beekeeping. Basically, you take a strip of hard plastic and stick it in the entrance of the hives. The bees have to walk over it to get in and out. And then you make a sort of paste out of beeswax and like wintergreen essential oils. I don't remember exactly what. And then you spread it on the plastic. So each time the bees want to get in and out of the hive, they have to walk through this miticidal goo.
Okay.
And then they try to get it off themselves and spread it all over their bodies.
Yeah, I mean, it definitely sounds like that could be helpful. Again, the scientist in me just wants to test it and have data and hard numbers to like say for sure. But yeah, I mean, I think I've seen other products too, where it's like I think it's called like a varroa gate or something, where the bees have to walk through this little like gated channel that's full of miticide. And, I think it's the exact same idea where you're trying to knock off any mites that are being brought into the colony that way. And so, yeah, I think anytime we're thinking about slowing down bees, as they're entering colonies and giving guard bees more of a chance to defend unwanted visitors, or about removing mites from bees as they come in. I think all of those things can be helpful for sure.
Ryan, your question had kind of a long part to it. Would you mind just unmuting and asking her your question?
Yeah, sorry about that. Okay, so basically, I'll just read it. So as I understand it, a mite bomb is when a colony collapses from a critical mite load and is then subsequently, because they're too weak to defend themselves, they are subsequently robbed. And then that critical mite load is transferred to the robbing bees. So what I'm wondering is, were the high mite load colonies in your experiment allowed to collapse and then be robbed out to test, what happens when a mite bomb detonates, right? When it collapses, it gets robbed. And if not, then it sounds like maybe the experiment was testing more mite drift between active colonies that have not collapsed yet, testing drift between high and low mite load colonies. And if that's the case, then was the experiment actually testing the effects of mite bombs or drift?
Sure. Yeah, that's a really good question. And so, yeah, I think I have like a slightly different definition of mite bomb, which is, you know, the one I gave where the actual high mite bees are going out into the landscape. And so I think that the term mite bomb has now started to be used more to describe the situation you're talking about. And I think that's because most of the research on the topic has shown that there's really never a case where a high mite colony is sending its own bees out into the landscape. It's almost always an issue of other colonies robbing out a high mite colony. And so I think, you know, you're absolutely right about that. And the actual original goal of the study was to try to get those high mite colonies to collapse. And you know, those high mite colonies had mite loads like over 10 mites per 100 bees. So really, really high mite loads. And I was like, great, this will be awesome. These colonies will be dead in like 3 weeks. It'll be perfect. And they just clung to life for, I don't remember the exact number of weeks, but it was like months. It was like more than 2 months that they clung to life, and they kind of slowly dwindled in population. Eventually, kind of in the last 2 weeks, they did sort of start to taper off in population. But yeah, I did really want to look at what happens when those colonies collapse really quickly. And that just did not end up happening for this experiment. And I definitely agree that I think that that should be done and is definitely like an important component to understand what happens when, you know, that super quick collapse does happen, because I think that is like a different situation biologically as well. So yeah, super interesting. Does that answer your question?
Yeah, it does.
Okay.
Thanks for answering it. And yeah, I mean, I guess I can see how it would be really difficult to run an experiment and just hope that they collapse at the right time, or
yeah, it's like, you know,
hard to
every
control.
time you're trying to keep bees alive, they'll just die on you for no reason. And then the one time I need them to die quickly, they just cling to life for weeks and weeks and weeks. And so yeah, I would like to try it again and actually get some to collapse, but it's hard to
Cool.
plan for. Yeah.
So,
Great
so you're
question.
planning to try to test that in the future?
Yeah. So, you know, as a postdoc, a lot of what you do is just experiments that your supervisor is already working on. So I don't have a lot of like creative license here to do things like that. So I'm hoping that once I get my like permanent real job, I can kind of get back into this field of sort of this inter-apiary mite transmission a little bit more and kind of get back to it.
Awesome. Cool. I hope you get to test that.
Thank you.
I think it'd
Yeah.
be really interesting data.
Yeah,
So
for sure. I agree.
Okay, we have another question. Is there any sense of whether best practices have any effect on how often a colony is lost from bees exiting the hive by absconding or swarming?
Yeah, good question. So, you know, I think specifically the 4 best practices I talked about today aren't necessarily designed to help with swarming. I think in general, a best practice to avoid swarming is definitely splitting your colonies at the right time. And so, you know, splitting has lots of other benefits as well. You kind of create a brood break, which is good for interrupting mite reproduction, which can kind of help your colonies start off with a lower mite load, which can carry over the whole season. So I think that there are definitely best practices that can help prevent swarming. Absconding is really like totally a different issue from swarming. You know, that means something else is kind of wrong with that hive, and that tends to be like a pretty specific case by case basis. So yeah, hope that answers that question.
And I personally had a question. So what can people do if maybe say you have a neighbor who doesn't treat for mites, or maybe you don't know if you have a neighbor who treats for mites or not. If you keep up on your own mite treatments, is that good enough, or is there something else to
Yeah, great question. And so, it's obviously pretty touchy because people who don't treat for mites like really don't want to treat for mites. So it's not like you could just go over there and knock on their door and be like, hey, like you need to treat. So I think, in most cases, we don't have that much control over our neighbors' actions anyway, unless, they're already coming to meetings and clubs and things like this. Or maybe they're just genuinely unaware, in which case, education could help. But yeah, I do think staying on top of your own mite loads is enough. Monitoring, I cannot stress how critical it is to sample your colonies after you have completed a treatment, because we see time and time again, and we've done it ourselves many times too, where you like put a treatment on and then you just kind of feel like you did your duty and you're good to go. But if you actually take a sample, 4 or 5 weeks after you applied that treatment, your mite load might be a lot higher than you think or than you expect it to be right after you treated. So just really, really make sure you're checking frequently what your mite loads are so that there's no surprises. And I think, the earlier you start in the season, the better. Kind of as soon as it's warm enough to sample, you should start because, again, those mites just kind of naturally reproduce all summer. And so if you can start your colonies off from really clean slate, then there's fewer mites in there to start reproducing in the first place. So yeah, now's the time to start.
Excellent, thank you. Somebody put in the chat, question for members. Would you be open to answering even more questions in our annual Pacific Northwest honeybee survey that could capture some of these variables? I'm not sure how we would respond to that, or how do we get a hold of that Pacific Northwest honeybee survey? Is Janae still here? Okay.
Jana, this is Dewey.
Oh, hi, Dewey.
And Danae and I work on that survey, and it is hosted by the Pacific Northwest, by the pub group. And so Janae's question is, so we have a series of questions, and we try to reach a point where we get enough people that will respond to it. And so Janae is asking if it was more specifically drilled down questions, would individuals be willing to try to answer those questions? So we'd love to hear an expression on that. It comes down to, we have to sort of in a broad stroke ask questions about feeding, about winterizing, about mite control. And we always are then asked specific questions when we give the talks of, well, what if this was the case, or have you thought of a question about that? And so we're always trying to refine the survey to try to make it of service. And since it is pub hosted, it is very appropriate to try to get an expression from pub if they'd like to have it be more specific than the types of questions now asked.
Okay, excellent. Yeah, so if anyone is here in the meeting, please chime in the chat to let us know if you'd be willing and interested to answer some more specific questions. And Dewey, you and I can catch up afterwards if you want me to put something in the email that goes out with the recording to the membership. I can do that too.
Okay, good.
Okay, so we have another question. Using oxalic dribble in early and late spring in addition to an early winter dribble have a downside?
I don't think it has a downside. I mean, I guess there's always hypothetical downsides of, applying pesticides to our colonies and sublethal effects and things like that. But I think, spring oxalic drip is a great way to start your bees off well on a good foot in the spring for sure. Yeah, I don't think there's any downside so large that it outweighs the upsides.
Excellent, thank you.
Great to do on packages too, before there's any brood.
Great. Do we have any other questions coming in last minute here?
Yeah, feel free to email me too if you think of anything later. I'm happy to answer emails.
That's great. That's a very generous offer of your time. Thank you.
Sure, no problem.
Well, it looks like we caught all the questions. So I really appreciate your time and all your dedicated information that you shared with us, and everything was wonderful.
Yeah, thank you so much for having me, and thanks everyone for listening and having such good questions. It's really my pleasure.
Thank you. Good night,
Thanks,
everyone.
everybody.
Hi, Kelly. Thank you. Great job.
Thanks, Dewey. Hope to see you soon.
Okay. Good luck with your bee collections tomorrow.
Thank you. Thanks. Bye.
Hey, Christina, I see you have a question on swarms that you
Yeah,
just swarm.
I just caught one today and it's
Oh, good for
cold,
you.
so I don't think I should move it into its box. I think I
I'm
should.
sorry, what did you do?
Should I move it into its box? Not tonight, right? It's too cold. Do it tomorrow?
I think you should probably do it tomorrow, but Dewey's still on here, so maybe Dewey would give us a second to answer that question.
Yeah, move them in tomorrow before they start all that, building all that comb. Good
Okay.
timing.
Got
Yeah,
it.
and we may get swarms tomorrow, folks.
There's one
Good
today.
weather. Yeah, one today. Tomorrow's even gonna be better weather.
Yeah,
Awesome. So excited. Thank you so much for the talk. It was a really great talk.
excellent. Thank you. All right, good night, everyone.